What's up, honors biologists? Welcome to Cell Signaling 101.
The best cell project you've seen yet.
There are 3 different types of cell communication. There's direct, there's local, and there's long distance. There are also 3 steps to cell communication, the first of which being reception, when the ligand binds to a receptor. If the ligand is small and nonpolar, like estrogen and testosterone, that means it can pass through the phospholipid bilayer and bind to a cytoplasmic receptor inside the cell. If it's larger, like dopamine and insulin, it has to bind to a membrane-bound receptor, like tyrosine kinase or a G-protein coupled receptor. When the ligand binds to the receptor, it fundamentally changes its shape and leads to transduction. The receptor has 3 parts: the intracellular, the hydrophobic, and the extracellular part.
After reception occurs and the ligand fundamentally changes the structure of the receptor, transduction occurs. This sets off a second messenger. Secondary messengers, in the case of dopamine, are G-protein alpha subunits, or in the case of insulin, tyrosine kinase. Can also include calcium ions or cyclic AMP. The second messengers cause a phosphorylation cascade in which phosphorylation occurs. This is when, for instance, ADP is changed into ATP or GDP is changed into GTP. After the phosphorylation cascade occurs, a cellular response is activated.
So, dopamine. Dopamine is one of the few specific ligands that's considered a neurotransmitter. Dopamine will start in the presynaptic nerve and will move across the synapse to the postsynaptic nerve. When this happens, dopamine will bond to the G-protein coupled receptor, and this bonding will cause the alpha subunit of the G-protein to move to an enzyme in the membrane. This enzyme causes the transformation from ATP to cAMP, or cyclical AMP, which will then phosphorylize or give energy to protein kinase. This protein kinase then moves to an ion channel, which lets the sodium and potassium concentration within the nerve to switch, causing depolarization, and therefore sending down the impulse of the reward system. In case you forgot anything about the nerves, you can review it back here on our back flap labeled Neuron Review.
Beta cells from the pancreas detect the high glucose levels in the blood and will release insulin. The insulin will then bind to the tyrosine kinase receptors of cells. ATP will phosphorylate the receptor, and the receptor will then phosphorylate protein kinase. Kinase will then take away the Rab protein, which stops GLUT4 from allowing glucose to enter the cell. Once the Rab proteins are removed, the GLUT4 channels open and glucose then enters the cell.
Hey guys, welcome to the YouTube video.
Hi, welcome to my brain.
Welcome to my brain.
I'm so dumb, I can't do this.
Is that right?
Just keep jumping up and down, yeah.
Okay, Rab removed.
Letting sugar into my body.
Phosphorylates.
You know what?